Welcome back to Unit 4, Lesson 2, Part 2, where we're picking up talking about the eukaryotic cell and its organelles. Previously, we talked about the nucleus and the ER, and now we're going to talk about the Golgi. The Golgi is this apparatus right here. It's a series of flattened sacs or stacks that contain fluid and proteins and enzymes and a variety of materials in their center. And at the ends they have these little spherical things called vesicles. So if we look at it under the electron microscope, they're these little sacs. And what makes them different, besides function, structurally different from the ER, is that each of these sacs are independent. They're not interconnected like the ER tubes are. And then off the ends, they're easily recognizable because they'll have vesicles off the end of them. Okay. If you look at this picture, there's something called the cis phase and the trans phase. Okay. And we'll talk about that in a second. So this is a electron micrograph. So if we're to take a closer look at the Golgi structure, it's a series of flattened membranous sacs. Usually about 6 or so of these sacs are stacked one on top of each other. And I said they had the cis and trans phase, so what I'm going to try and do is draw for you an example of the Golgi. Okay. And the tough thing here is going to be to color in this portion. So what I'm coloring in black now is to indicate that's sort of the internal region where a series of events, reactions, proteins occur. Okay. And maybe I'll draw all the stacks like that first. And then we'll add in the three-dimensional aspect of it. Or my attempt of 3D. So we'll just draw 5 for this purpose. So these are series of vacuoles that have been flattened. They sort of line up one above the other. And in that above picture, there was a comment that you had a cis side which is closest to the ER / nucleus. And then you have the trans side, which you could say either is farthest from the ER nucleus or closest to the plasma membrane. And in the middle, if any of you have had the misfortune of having an MCL injury in your knee, you should know MCL stands for medial collateral ligament, and the M for middle, it's called the medial stacks, because it's not really a side, it's in the middle, equals middle. And it's important to distinguish cis side from trans side because different things occur at these locations. Now, the really cool thing that coming off of these various stacks, you get little things that come off of them. And I'm drawing them not to scale, they're a little big here, but they're little round spheres. It's me trying to represent the little round spheres. And they come off, and then they can merge back onto them and transport things from one end to the other. And these are called vesicles. And this process of coming off is one of my favorite words in biology. It's called blebbing. And that's the vesicle moving apart from whatever membrane it started at. It's just fun to say, blebbing, or bleb off. It's a good word. Okay? So, that's the structure of the Golgi. Now we're going to look at the function. So, the first thing are that as proteins move through, they're modified. So proteins are modified as they move from cis to trans Golgi. I really should capitalize Golgi since it's named after the person who discovered them. And by modified, I mean we can do things like add or alter carbohydrate chains that are attached to them. And these chains help dictate the function of the protein. As well, by the time they reach cis Golgi, cis Golgi helps to sort the proteins into vesicles destined for one of three locations. And if you think back, all the proteins that enter the Golgi are those same ones that entered the ER, and we said proteins would enter the ER depending on them going to one of three locations. So it's the same idea again. The first could be if they were targeted to a particular organelle, such as a lysosome. They could be packaged into a different vesicle that might be destined for the plasma membrane. or they could be packaged in a similar vesicle that is destined for the plasma membrane, but as it fuses, it actually secretes the protein. So secretion out of the cell. And there's an interesting thing. It's clear that proteins move through the Golgi, become mature, and become sorted. But what's not sure is how the proteins move. This is unclear if proteins move from cis to trans, strictly via vesicle, so they get packaged in the vesicle and it moves forward to the next stack and so forth, which is called the vesicular model. Or if the entire stacks moves forward cis to trans, carrying the protein with it, and that the vesicles are really just recycling things back to the earlier phases. That one's called the maturation model. Or what looks more likely, whether it is a combination of the two processes. And the reason scientists are interested in this is because there are defects, illnesses that arise when proteins don't move properly through the Golgi. And they're trying to figure out how is it supposed to work normally so we can understand how it fails to work sometimes. Okay. Now if we go back to our analogy, we were talking about the cell as a car factory. So the Golgi's job is to help move proteins along, make some additions to them, and sort them to the correct locations in the cell or beyond the cell. So if the Golgi is part of that car factory, then the Golgi is like part of the production line. Where final options are added to the car. And it is sorted for shipment to various dealerships. Based on its options. Example, if you added in the AC unit into the car, yeah, it'd probably be coming to a dealership where it's very warm. So somewhere like Kelowna as opposed to somewhere like Alaska. Okay, So that's the role of the Golgi. Next, we're going to take a closer look at these vesicles. So, in this diagram, vesicles are really quite difficult to see. They're the smaller structures. All these little tiny things. This could represent a vesicle, though you'd expect it to be a little more spherical than tube-like. These little guys could represent vesicles. Okay. So if we're looking at their structure, they are membranous spheres, typically spherical. That vary in size. But are very small in most animal cells. So they're the second smallest organelle we'll talk about, ribosomes being the smallest. There are some that are just as small in diameter but much bigger in length. And there's really two categories of vesicles based on function. First we have one, constitutive, secretory, or secretory, depending who's saying it, vesicles. If you do something constitutively, it means you do it automatically or all the time. So these continually fuse with plasma membrane to secrete their contents out of cell, or to add lipids and protein to the plasma membrane. And it's the latter part of that that is more common for constitutive vesicles, in that they are delivering lipids to the membrane so that the cell membrane can actually grow. So if you look back up here at the diagram, this is an example of constitutive secretion and constitutive vesicles. So here's the constitutive vesicle. It's blebbing off the Golgi. Gotta love that word, bleb. And then it's moving, and we'll talk about how it does that later. It's moving up to the plasma membrane, where it fuses and becomes part of the plasma membrane. And anything inside it that's dissolved in its liquid is secreted out, so those are secreted. And anything attached or embedded in its membrane becomes part of the plasma membrane. And this process is called exocytosis. Exo meaning, you've got it, exit or out. Cytosis, or cyto, equals cell or out of the cytosol. Okay? So exocytosis is the process of sending something outside of the cell. And in this case, it's constitutive because it happens without any sort of signal. It's a default mechanism. Now, if you look at the picture carefully, you can see what our other form is. We have constitutive secretory vesicles, and we have regulated secretory or secretory. Secretory is actually correct. Vesicles. Okay. And these only fuse with plasma membrane in response to a signal from the external environment. Such as release of insulin, digestive hormones, digestive enzymes, and neurotransmitters, which we will talk about when we get to the various systems. So these both are exocytic vesicles, if we were to give it a broader class. And it's just what causes them to release things that differ. Okay, so if we go back up to the picture, we can see how what happens first is there's some sort of signal here, and it binds to a receptor, which is showing it doing that over here. That sends a signal into the cell telling the vesicle to fuse. So 1, 2, 3. Okay. Now, because I know you guys are brilliant, and we have exocytic vesicles that move things out of the cell, what other type of vesicles do you think we also have? Yes, I know I could trust you to be brilliant. The opposite of exo. We also have endocytic vesicles. Which take things up into the cell. And we'll talk more about that next day when we're going to talk about how we regulate what comes in and out of the cell. And the last type of vesicle we have is called, when we're not talking about exiting or entering the cell, is called transport vesicles. and these move lipids and proteins between various organelles in the cell. Okay. And if we're going to go back and do the cell is a car factory analogy, then vesicles are like crates used to bring various components into, out of, and throughout the factory. So we put things in them and we move them around. Okay. Next, we're going to talk about vacuoles. So vacuoles are not always easy to see or distinguish in animal cells just because they're about the size of several other objects within the animal cell. But we are going to term this as an example of the vacuole. And you'll notice there's more of these, but I'm not going to color them because we're going to use them for different things. There's several things that are about the same size, but they perform different functions. And if you're going to tell from a picture what they are, I'm going to have to give you more information. So, in an animal cell, these vacuoles are pretty tiny. In a plant cell, they can be as big or even bigger than the nucleus. They can sometimes be the biggest structure. So remind you that this is a plant cell vacuole, whereas this is an animal cell vacuole, much smaller in comparison. But in an animal cell you might have several vacuoles, whereas in a plant cell you have fewer. So we're going to talk about their structure. Well, as you've learned, they vary depending on the type of cell you're in, but we're going to focus on human cells. So they are membrane sacs that are larger than vesicles, and often comparable to two other organelles we're going to learn: lysosomes and peroxisomes in size, but not in content or function. And that's how in a picture you would know them apart, because I'd have to give you a hint about either what's in them or what they do. And we'll just say in animal cells. In terms of their function, in animals they do three things. Or three key things. they store nutrients, water, and waste. In plants, they do those three things. They do one thing, they store, but they store nutrients, water, waste, and because the plants will go green, and they store toxins that make the plant less edible, so things that protect the plant from being eaten by other things, and pigments that control the plant color. So it's not very complicated what vacuoles do. They're pretty much a storage space, and they can be used to store a variety of items. So if it was the cell is like a car factory, then the vacuoles are like storerooms, cupboards, and drawers. Any organized space where you would store things within the factory. Okay, so we're just going to stop there with this section. If you have any questions about that because you missed class and you're watching this at home, please come see me and I will try and answer your questions. Until then, have a good day. Hope to see you in class.